Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today, in the series of videos lecture, we are going to talk about B cell biology in detail. In the previous series of videos lecture, if you guys remember, we talked about the T cell biology in detail. And in T cell biology, we talked about the overview, we talked about T cell receptor, we talked about types of T cell. And types of T cell, we talked about helper T cell in detail, cytotoxic T cell, T regulatory cell, and types of helper T cell also. Okay, and after that, then we talked about the T cell activation, T cell differentiation, T cell maturation, and T cell development. So all these things we talked about in T cell biology in detail. So if you miss these videos lecture, this series of videos lecture, so go back to my channel and watch this video lecture, because it is also important to know about the adaptive immune system in which the T cell play an important role. In this video, we are going to talk about another adaptive immune system part, which is actually the B cell biology in detail. Okay, so without wasting any time, let's start the video. Okay guys, so now let me write. Okay, B cell biology, or it means that the life cycle and the life history or the story of the B cell. Okay, just like T cell, we talked that if we are going to talk about the T cell activation, maturation, their structure, their activity, their types, each and everything, whenever we are going to talk about, then it is called a T cell biology. Similarly, in case of B cell biology, whenever we are going to talk about B cell biology in detail, for example, whenever we are going to talk about the B cell activation, whenever we are going to talk about B cell development, B cell maturation, B cell structure, B cell differentiation, each and everything, whenever we are going to talk about the life, the cycle, the history and the background, and means the discovery of the B cell in detail and the study of the B cell, whenever we are going to talk about, then it is called as B cell biology. So if again I write here, so what is actually B cell biology? So in B cell biology, we mainly talked about the introduction. Introduction means the basic overview of the B cell biology. We are going to talk about the structure of B cell, and we have in B cell biology we have B cell activation, their differentiation, their maturation, and development. So all these things, whenever we are going to talk about all these things in detail, not only these things, whenever we are going to talk about some other things which are related to B cell, it will be called as B cell biology. So in this first video, we are actually going to talk about the B cell biology overview, means the introduction of the B cell in detail, the concept behind the B cell, or the big picture of the B cell, or the summary of the B cell biology. Okay, this B cell is also called as B lymphocyte. So let me write. Okay, the name B cell. First of all, let me a little bit talk about their discovery, that how actually it discovered. The B cell is first of all originated, or it was first of all discovered inside the bursa fabrics of the bird. Okay, or we can say it was first of all discovered inside the bone marrow of the human body, and after that the name was given B cell. Why? Because B means bursa fabrics, or B means bone marrow or bird. That's why the name is given B cell. Okay. It is a little bit about the B cell discovery. Then we have, the structure of the B cell. So if we see the structure of the B cell, so it's mainly, not exact circular. The B cell is not exact circular in structure, but somehow it's similar to circular in shape. Whenever we are going to talk about the B cell structure, so mainly it have the receptors. Mainly we identify the B cell by its receptor, which we call that B cell receptor. So let me draw. So, and moreover, this B cell have all these receptor, and they have also the nucleus portion, as you can see, and inside the nucleus they have the genetic material, which is the DNA. Okay, so this is actually the B cell structure. Mainly, most of the time, whenever we talked about the B cell structure, so they have all these receptor, which is actually similar to the antibody, as you can see. This receptor is specially responsible for the B cell activation. We will talk it later. And moreover, they have also a genetic material and some other organelles as well, because this is the eukaryotic cell. So just imagine that they have also the other organelles, mitochondria, Golgi body, each and everything. Okay. So this is actually the structure of the B cell. So now let's a little bit talk about the B cell receptor, which is very important. B cell receptor are mainly formed from this DNA portion. Inside the DNA there are some region which are specially responsible for the B cell receptor. So B cell receptor are mainly formed by this DNA. So there are some region due to which transcription translation occur, and it will make the different types of B cell receptor. There are almost 50,000 different types of receptor on the surface of the B cell. So it means that they will identify different types of microbes through their different epitopes. So we can say that almost we have 50,000 different types of B cell receptor, and almost we have 10,000 variation combination. What does it mean? It means that we have different types of microbes, like viruses. So it is also organized by the B cell receptor, like fungi, like bacteria, different types of bacteria, different types of viruses having different types of epitope. So all the microbes which are organized, there are thousands of microbes which are surrounding. So all these microbes are actually recognized by this B cell receptor, because there are different types of the B cell receptor on their surface. So on the other hand, this B cell receptor, let me write BCR, this B cell receptor is also called as membrane bounded microglobulin. Why? Because it is similar to immunoglobulin, means antibody. Okay, so this is a little bit talking about the BCR, B cell receptor. So now let's talk about the B cell activation. So mainly the B cell will be activated inside, when there is the microbes internalized, let's suppose any kinds of pathogen which come contact with this antibody. Just imagine if there is any kind of antigen comes inside the body. So then what happen? They will, contact with this portion, or this antigen portion, antibody portion, as you can see. So now remember guys, when there is any kinds of bacterial cell microbes get internalized inside the body, so then when it contact with this receptor of the B cell, so then what happen? It will provide signal to the nucleus, and then the nucleus will decide that what need to do against this pathogen. Okay, they actually recognize the epitope of the pathogen, means let's suppose this portion is the epitope. Okay, so once the epitope portion is recognized by the paratope of the antibody, so after that antibody or receptor of the B cell, so after that what happen? It provides signal to the nucleus. Then the nucleus, what actually the nucleus will do? Nucleus will, inside the nucleus there are genetic material. The genetic material are rearrange themselves and start transcribing and translating the DNA or the RNA molecules. After that what happen? We get different clone of the B cell, different copies of the B cell, as you can see. So let's suppose these are what? These are the B cell. Okay. But remember guys, among each B cell, what actually we have, among each B cells, there are some B cells which we call that memory B cell, as you can see. And there are some cells which we call that effector B cell. Okay. You know, I already explained the memory B cell also, but let me once again tell you, the B cell which are, make their copies, among them we have memory B cell. So the memory B cell are those cells which can store the information of the antigen or the pathogen. They can store all the activity of that pathogen, that what kind of damages they can produce or they produce inside the body. Due to that way, once they store the information, so then it will be ready, and then they will, attack to this one pathogen at the second time fastly. So let's suppose if the first time the microbes comes, so then we get the memory B cell, they store all the information. If the second time the same microbes come, so all the memory B cell is already present. After that, what actually these memory B cell will do, although they know the properties of this pathogen and they know the activity, that what kind of damages they can do, so then these memory B cell, without any thinking, they will directly kill this pathogen fastly as compared to the previous attack. So the first attack, the killing of the first attack by the B cell is very slow, but the killing of the second the killing of this second of the same pathogen by the second time, it's very fast. Why? Because they know everything. So that's why it will kill it so fastly. This is the unique properties of the memory B cell. Then we have effector B cell. And what actually effector B cell can do? Effector B cell can produce different kinds of cytokines, as you can see. They will produce different kinds of the cytokines. Each cytokines will activate, phagocytic cells. Phagocytic cell. Among phagocytic cell we have neutrophils. Let me write. We have neutrophils, we have macrophages, and we have dendritic cell. Dendritic cells. What actually each cell can do? Each cell will engulf the microbes or the pathogens which can cause damaging inside the body. So once each cells are activated, so then they will engulf it. Sometimes these effector cell will convert the B cell into another cell which we call that plasma cell. So after that we get another cell. This is about the plasma cell. Plasma cell is a little bit in large in size. There it is a little bit large in size than the B cell, and moreover this plasma cell will produce those antibody which are against this one antigen. If this antigen require IgG antibody, so this plasma cell will produce IgG antibody. If this cell required IgM antibody for the killing, so then they will produce IgM antibody. Similarly, if this antigen required IgA antibody for the killing, so then they will produce IgA antibody. So almost we have 5 types of antibody, like IgA, IgM, IgG, Ig. These types of the antibody. So I forget the last antibody name, but we have almost 5 antibodies. So whatever the antigen needed antibody for the killing, so then this plasma cell will produce those kinds of the antibody. So we get a point that plasma cell is actually what? This is actually a kind of antibody factory which can produce different types of the antibody against this pathogen. Normally what happen, this kinds of phagocytic cell will engulf it. but sometimes the phagocytic cell are failed to engulf this antigen. Then what happened after that? Let's suppose this is let me draw once again the antigen here, although there is a lot of antigen. So let me draw another antigen there. So if this microbes or pathogenic microbes is not engulfed by this phagocytic cell, so then this plasma cell will secrete antibody which are against this antigen. So then the antibody coat this microbes, as you can see. So now these microbes are become coated. What happens sometime, this antibody by itself kill the microbes alone. Okay, sometimes these antibody are also failed to kill this. Then what happens, they will secrete, or they will call the phagocytic cell that now this means the bacterial cell or microbes are become weak, because we weakened it by ourselves, means it is weakened by these antibody. So after that this phagocytic cell will easily engulf it. Okay, or in the simple language we can say that if the antibody are coated, so it become tasty for the phagocytic cell. So phagocytic cell can easily engulf it. Okay, but without antibody coated, then it will be very easily for it will be very difficult for the phagocytic cell to engulf it. That's why coated are very important, means this microbes coating are very important. Sometimes these microbes are coated with the complement protein which can coat this, and due to this way the phagocytic cell can also engulf it. Sometimes this complement protein will make a pores, due to which there will be no need of the phagocytic cell to engulf it. So due to the pores the microbes will be killed. Okay, so what happened, we get a point that sometimes this microbes is killed by the phagocytic cell, means phagocytic cell will engulf it. Sometimes this microbes is killed by the antibody which are produced by the plasma cell, and sometimes this microbes is killed by the combination of both phagocytic cell as well as this, what we can say, this antibody. Means let's suppose this is the phagocytic cell, so phagocytic cell will engulf it. Let's suppose this is the macrophages, they will easily engulf it. Okay, so this is how we can say that the B cell are activated, and then it will be differentiated into memory B cell, effector B cell, and plasma cells. Then we have the B cell development and their maturation. So mainly the B cell is developed inside the bone marrow. Okay, let's suppose this is what, this is the bone marrow. Okay, inside this bone marrow we have different kinds of immature cell, like we have the most important cell, we have the hematopoietic stem cells. Hematopoietic stem cells, or we have a stem cell that give rise different kinds of the cell. So here I will recommend you that if you want to know about the B cell development and their originated and their formation, you need to watch the hematopoiesis process, which I already explained. You can go back and watch that video lecture. Okay, so what in hematopoiesis, what we talked that we already talked that the B cells are mainly developed inside the bone marrow. Inside the bone marrow we have a stem cell which we call that hematopoietic stem cell. From hematopoietic stem cell there are different types of the cells are formed, and from each steps of the cell then finally we get a B cell. We will explain it later in the B cell development in detail, but this is how the B cell are mainly developed inside this bone marrow. Okay, that's why the name is given B cell. And if we talked about their maturation, so again it will be mature inside this bone marrow. Okay, so what we get a point that for the maturation and development of the B cell, it required the bone marrow. And in the T cell, if you guys remember, we talked for the development of the T cell, it required the bone marrow, but for the maturation, it required the thymus. That's why the name is given T cell, means thymus. Okay, so this is all about the B cell maturation. Now, if we talked about the B cell maturation, so remember guys, B cell mature inside the bone marrow. There are the neutrophils. Neutrophils actually mature the B cell. And in simple language, we can say that neutrophils are actually the teachers of the B cell. They actually guide the B cell that what you need to do when you have function when you have DVT inside the bloodstream, or if there is disease or condition, then what you need to do. So all the activity, all the knowledge is provided to the B cell by the neutrophil. Neutrophil is actually the teacher of the B cell. So this is all about the B cell maturation. Okay guys, and another important thing is that about the B cell, that mainly the B cell are involved in the humoral immunity. Humoral immune. What does it mean? It means that inside the humoral immunity we have the antibody and we have the B cells. So we get a point that B cell are always involved inside the humoral immunity. Okay guys, so this is all about the B cell activation. This is all about the big picture or the concept behind the B cell biology. So it's very important for you guys to know about the B cell biology in detail. Once you get idea about that, so then the rest of the video which are regarding to the B cell will be very easy. Okay, inshallah in the next video lecture we are going to talk about B cell receptor in detail. Okay, so please stay tuned and keep watching. If still you didn't get idea about that video lecture, so you can write your question inside the comment. I will try my best to give you answer as soon as possible. Thank you for watching this video.